Hello, my name is Kristen Pisani, and I am a master's student at the University of Georgia in the Animal and Dairy Sciences Department. Today I'm going to be talking to you about the ruminant microbiome when dieting really matters. So basically, we're going to be talking about the impact of diet on the microbiome and what effect that can have on you, the producer. So, just a little basics that I'm sure a lot of people already know. Um, Within the rumen, it has its own vast pool of microorganisms, this whole ecosystem full of bacteria, archaea, fungi, protozoa, each having their own role within that system, which I'll get into a little more about that in just a minute. But each of those have their own primary role within the rumen, and it's really important that we have all of those because they do all significantly contribute to the degradation of feedstuffs and providing energy sources to the animal. We always have to remember that when we have ruminant animals like cattle and sheep and goats, we are feeding that microbiome before we're actually feeding our animal. So, some impacts on this microbial population include health status. So, if they're not feeling well, that can impact that. Geographical location, species, sex, and what we're going to talk about is diet. So, the diet can have a huge impact. As I said earlier, we are feeding that microbiome before we're feeding our animal. So, it makes sense that it would have a huge impact. I got into a little bit of how each of those microbes that I showed on the last slide had their own roles. So, I have up on here Butyrivibrio, fibrosolvens, Fibrobacter succogenes, and Ruminococcus bromi. Those are just some examples. Obviously, you just looking at them isn't going to really mean anything to you, but we know that the first two, the Butyrivibrio and the Fibrobacter, are fiber degrading bacteria. So their role is specifically breaking down fiber that comes in, so cellulose, hemicellulose, lignin, those kinds of things. They each have their own substrates that they'll use for their energy, and then their products might be a substrate for another bacteria or archaea within the microbiome. Whereas Ruminococcus bromi is a known starch degrading bacteria, and how these impact each other and how it really impacts the microbiome is Ruminococcus bromi and other starch degrading bacteria typically have products such as lactate, which leads to lactic acid. As indicated by the name, lactic acid is going to decrease the pH of the rumen and can lead to disorders like acidosis. Acidosis is just low pH in the stomach, in the rumen, which will cause your animal not to eat. It can affect their performance. So, really, focusing on diet is super important and can have a huge impact on your animal's health and their performance. So, when I say performance in my head, if you don't know me super well, you don't know that my project is residual feed intake essentially. So, I'm going to talk about these impacts on residual feed intake. So, residual feed intake, or RFI, is a measure of energetic efficiency. It's defined as the difference between an animal's actual feed intake and their predicted feed intake. And that prediction is based on their rate of gain and body size. So, if you look at that graph on the right, you can see it references average daily gain as well as their body size. And then there's expected feed intake and observed feed intake. Those animals that are below that solid line have a low RFI, and those that are above have a high RFI. So, meaning they have a low residual feed intake. So, that means they intook less feed than we had expected them to grow at the same rate. That means that an animal can eat less feed and still grow just as well, whereas a highly efficient animal is growing just as we expected them to, but is intaking more feed than we expected them to. So that means that that animal is less efficient. A low RFI animal would be more efficient. Obviously, we want our animals to intake less than we expected them to. That's going to decrease our input costs and is really important to producers. I'll get into that a little bit later. But RFI specifically is super useful. We have a lot of different measures of efficiency. There's feed to gain ratio, FCR. So Why RFI specifically is because it's moderately heritable, so we can kind of track it through genetics, but more importantly, it's independent of growth and body size. So by that I mean we can selectively breed for RFI, low RFI, high efficiency, but so far there's been no increased body size from that, no increased average daily gain, which is actually good because that means that we're not going to increase our input costs or create a larger frame size animal that in turn might have more issues in calving, would require more inputs, more land. So residual feed intake is become a really hot topic just because of how useful it is. So because we know that the microbiome has an effect nutritionally on our animal, it makes sense to look at how that might affect efficiency because we're talking about feed, we're talking about a microbial population that first touches this feed before it gets to our animal. So this is an area that is starting to get explored a little bit more. This one study that I had found by Carberry in 2012 is almost exactly what I'm doing for my project. A little bit different, actually a lot of bit different, but still it's kind of a basis. They fed a high forage diet and a low forage diet to animals that were divergently selected for RFI. So we have a group of high RFI animals and a group of low RFI animals, and both were fed a high forage diet and a low forage diet. And there was a difference in the rumen fluid of those animals. So if you look in that picture on the right, you'll see there's the high forage diet on the left and the low forage diet on the right. Those black dots are our, low RFI animals, so high efficiency. Anything in black is high efficiency, and anything clear or no color is low efficiency. And you can kind of see that those have grouped together. I don't think you can see my pointer on the screen, but you can see some grouping there. So there is a difference, and it was significant in this study. And they related that back to a difference in Ruminococcus albus, which is also a fiber-degrading bacteria. So in low RFI animals, high efficiency, there was a 1.7-fold greater influence of Ruminococcus albus. So there was 1.7-fold more in low RFI animals than in high RFI. So that's just one example of how the microbiome can have a connection with RFI. And this is found in some of other studies that some re-ranking of animals can occur when diets are varied. So high efficient animals might become low efficient on different diets, and it's just something that needs to get studied further. Enter me, because that's what my project is, so stay tuned for that. Hopefully I'll have some good data and we can talk a little bit more about it, but overall we can potentially use this microbiome and manipulate it to create more efficient animals and therefore decrease feed input costs and increase our profits overall. Here are a list of my references. Thank you so much for listening, and if anyone has any questions, you can ask Dr. Calloway for my contact information, and I would be happy to discuss this further with you. Thank you.